Now second pathway is our C4 pathway. Why it is called C4 pathway? Already I tell you, because the first product, it is 4 carbon product, and it is OAA, oxaloacetic acid. That's why it is called C4 pathway. In C3 pathway, first product is our 3PGA, and this 3PGA, it is 3 carbon molecule. That's why it is called as C3 cycle. Do you understand? This pathway, C4 pathway, it is discovered by Hatch and Slack. That's why it is also called as Hatch-Slack pathway. Hatch and SLACK, Slack pathway. Hatch and Slack pathway, right? Plants that are adapted to dry tropical region have the C4 pathway. Dry means here what happens? Here what happens? Here water, that is availability is less. So they adapt themselves. That means what? Here they have more efficiency to fix the carbon dioxide than C3 plants, right? We can say twice, because they use half water to fix the same amount of carbon dioxide in C3 plants. They use half of water, means they have what? Dry tropical region. They have the C4 pathway mentioned earlier. Though these plants have C4, that is oxaloacetic acid. It is the first stable product, right? First CO2 fixation product. They use the C3 pathway, or in Calvin cycle. Look here, these plants have C4 oxaloacetic acid. It is the stable structure, first product. And C3 means here given C3 or Calvin cycle, it is our 3PGA, right? Here what I have? Here we have oxaloacetic acid as the main biochemical pathway. Look here, C4, it always use C3 because I already tell you C3 is universal, right? Then in what way are they different from C3 plant? Morphologically, anatomically, they are also different. This is a question that you may reasonably ask. So C4 plants are special. Why they are special? Because some specialty is there. What is specialty? They have a special type of leaf anatomy, and this leaf anatomy is called Cranj anatomy. CRANZ, right? Cranj anatomy. Cranj anatomy means what? Suppose that this one is our vascular bundles. So they have this bundle sheath cells. Bundle sheath cells. These are bundle sheath cells. Okay, bundle sheath cells. Look here, these bundle sheath cells. On the outside, this one is our mesophyll cells. These are mesophyll cells, right? So these mesophyll is attached with bundle sheath cells through plasmodesmata. Through plasmodesmata, they are attached. There is no in between. They have no, and that is intercellular space is absent. In case of this bundle sheath cell, intercellular space is absent, right? This is our vascular bundle. That's why they are in a Cranj anatomy, means in a concentric ring-like structure is there, right? They have no interstellar space. First thing is there, they are airtight, right? Second, they are attached with the mesophyll cells through plasmodesmata, right? Third, here also dimorphic grana, we can say dimorphic grana is there. Dimorphic grana means what is here? Dimorphic chloroplast is there. CHLORO, chloroplast. Dimorphic chloroplast means what? Here, two types of chloroplast. First one is called granal chloroplast, and second one is called agranal chloroplast. Granal chloroplast means chloroplast in which grana is present, and it is present in mesophyll cells. Because mesophyll cells, they are present, this one, outside, near the sunlight. So they have grana, means light reaction is there. But chloroplast, which are big in size, large in number, these are agranal. And chloroplast which are small, right, they are granal. So chloroplast which are present in bundle sheath cells, they have no grana because they have only dark reaction, stroma reaction is there, right? Okay, look here, what is E1? They tolerate high temperature. Why they tolerate high temperature? Because they have a PPDK enzyme is there. What do you mean by PPDK enzyme? PPDK ka, what is the full form of this one? Its full form is pyruvate phosphate dikinase. It is pyruvate phosphate dikinase enzymes which is sensitive against low temperature. That's why in low temperature they shows less affinity, right? They show the response to high light intensities. They lack a process called photorespiration is absent here because in mesophyll cell Rubisco enzyme is absent. Here bundle seed cell they contains Rubisco, right? But in mesophyll cell they have no Rubisco but they have peptidase, right? That's why photorespiration is absent and have a greater productivity of biomass than C3 plants. Now let us understand by one by one. Study the vertical section of leaf, one of the C3 plant and other of the C4 plant. When we see the vertical that is diagram, then we understand in case of C3 plants we have palisade tissue, we have also spongy layers, and bundle seed cells are absent also. That is bundle seed. We cannot say bundle seeds absent. Seed cells are absent, but what happens? They are not involved in photosynthesis, right? Mesophyll cells means they involve in all the reactions. Dark reaction, light reaction occurs in mesophyll cells only. In case of C3 plant, but in case of C4 plant, what happens? Here bundle seeds. I already tell you this one. This two types of cells are there. Bundle seed cell and another one is called mesophyll cells are there. Right? So there is some difference which I tell you. In C4 plant, what happens? Bundle seed cell they have a granule chloroplast. Mesophylls they have granule chloroplast. Right? Bundle seed cell they have Rubisco enzyme, but mesophylls they have peptidase. Right? Peptidase full form is phosphoenol pyruvate carboxylase enzyme. Right? Because only carboxylase enzyme is there, oxygenase enzyme is not there. That's why here that is we can say photorespiration is absent due to the absence of oxygenase enzyme, right? But in C3 what happens? Rubisco is present in mesophyll cell also. So Rubisco present with is also oxygenase enzyme. Rubisco it is double mentioned, carboxylase as well as oxygenase. When concentration of oxygen is more and temperature is also high, then what happens? Photorespiration occurs. That's why in C4 plant productivity is greater than C3 plants. Twice greater, we can say, twice greater, right? Study vertical section. Okay, vertical section means what? Here, do both have the same type of mesophylls? We can say no. In dicots, mesophyll means what? It is differentiated into palisade tissue and spongy tissue. But in case of monocots, C4 plants, only mesophyll is not divided into two parts. But when we anatomically say, so we know bundle seed cell are different and another mesophyll cell is different. Do they have similar cells around the vascular bundle sheath? No, they have no similar cells, right? Because cranch anatomy is present in only where C4 plants, and this anatomy is absent in C3 plants. The particularly large cell around the vascular bundle, look here, these large cells, vascular bundles of the C4 plants are called bundle sheath cells, large cells. But in case of C3 plants, we have no large cells. And the leaves which have such anatomy, and these leaves are called as cranch anatomy. Already I tell you, cranch means what? Wreath. Wreath means covering, right? Surrounding, and is a reflection of arrangement of cell. Why it is called cranch? The bundle sheath cell may form several layers around the vascular bundles, means more than one layer there. They are characterized by having large number of chloroplasts. Large number of chloroplasts, first thing, and these chloroplasts are also big in size. Second, thick wall which is impervious to gaseous exchange. They have thick wall, no gaseous exchange occurs, right? And no intercellular space is present. Three things is given here. First, large number of chloroplasts, and also big in size. And which type of chloroplast? They are agranal chloroplast. Second, thick wall which is impervious to gaseous exchange. Third one is no intercellular space, right? You may like to cut a section of leaves of C4 plant like maize or sorghums, amaranthus. They are also here, right? To observe the strange anatomy and the distribution of mesophyll cells. We can see this one, right? And we also did this one in our plant anatomy already, right? Now, it would be interesting for you to collect the leaves and their diverse species plant around you and cut vertical section of the leaves. It is for only practical, right? Observe under the microscope. Look for the bundle sheath around the vesicle. The presence of bundle sheath would help you identify they are C4 plants or they may be C3, because in C3 cycle the bundle sheath cells are absent, right? Now study the pathway shown in the figure. This is figure, this one is figure, right? So this study here we are going to read this one. This pathway that has been named the Hatch-Slack pathway because it is discovered by Hatch and Slack scientists, is again a cyclic process. Let us study the pathway by listing the steps, right? So look here what is given. The primary CO2 acceptor is a 3-carbon molecule, and this 3-carbon molecule is PEPcase. Full form is phosphopyruvate. That is what is given, phosphoenol pyruvate, right? Carboxylase, PEPcase, and is present in the mesophyll cell. Rubisco is absent. The enzyme responsible for this fixation is PEP carboxylase, means phosphoenol pyruvate carboxylase, right, or PEPcase. It is important to register that the mesophyll cell lack Rubisco, and that's why here photorespiration is absent. The C4 acid OAA is formed in the mesophyll cells, right? So OAA, oxaloacetic acid, which is a 4-carbon molecule, it is formed in the in mesophyll cells. Now this oxaloacetic acid, which is a 4-carbon molecule, it is converted into aspartic acid, as for example in sugarcane, or in malic acid, like in sugar sorghum, right? Like malic or aspartic acid in the mesophyll cell itself. Then what happens in mesophyll cell? First carbon fixation happens in mesophyll cells. Means what? PEP plus phosphoenol pyruvate and plus HCO3. I will explain you, right, how from where HCO3 is formed, right? When CO2 comes into the cytoplasm, it binds with water molecule. Now when it binds with a water molecule, it forms the carbonic acid, that is H2CO3. Now H2CO3 is broken down into H plus and HCO3 minus. This HCO3 minus, it binds with phosphoenol pyruvate and forms first molecule, that is oxaloacetic acid. Now this oxaloacetic acid, which is the 4-carbon molecule, it is converted into aspartic acid or malic acid. When it is converted to aspartic and malic acid, this aspartic acid or malic acid, it is transported through desmota, through plasmodesmata into the where that is our bundle sheath cells, right? Bundle sheath cell. In bundle sheath what happens? This aspartic or malic acid, through decarboxylation, it releases 1 carbon dioxide, and this 1 carbon dioxide which is released from this malic acid, right, it is used in C3 cycle. C3 cycle occurs in bundle sheath cells, right? And now this malic acid or aspartic acid, it is converted into pyruvic acid. When it is converted into pyruvic acid, it is just returned back to the mesophyll cells. And in mesophyll cells, this pyruvic acid is converted by using NADP. It is converted into phosphoenol pyruvate using a NADPH and also 2-ATP, right? So 2-ATP is used in the conversion of pyruvic acid to phosphoenol pyruvate. That's why here total number of ATP which is used in C4 cycle, it is 5, right? 3 in Calvin cycle, C3 cycle, which occurs inside the bundle sheath cell, and 2 in mesophyll cells in the conversion of pyruvic acid to phosphoenol pyruvate. Okay, look here. This is the total cycle. In then form malic acid, aspartic acid in the mesophyll cell itself, which is transported to the bundle sheath cell. When it converted to malic and or aspartic acid, it is transported to the mesophyll cell, right? Oh, sorry, bundle sheath cell. In the bundle sheath cell, these CO2 acids are broken down to release CO2 means through decarboxylation into CO2 and 3 carbon molecule, that is our pyruvic acid, right? This 3 carbon molecule is transported back, that is returned back to the mesophyll, where it is converted to PEP again, thus completing the cycle by using of 2 ATP, right? Here PEP again by using of 2 ATP, right? Thus complete the cycle. The CO2 released in the bundle seed cell enters the C3 cycle or the Calvin cycle. This is a very common to all plants, means it is universal. The bundle seed cell are rich in the enzyme of rubisco, that is ribulose bisphosphate carboxylase oxygenase enzyme, but lack PEP case. Mesophyll cell, they have PEP case but no rubisco. In bundle seed cell, they have rubisco but no PEP case, right? And in bundle seed cell, what happens here? No oxygen is very less, only carbon dioxide is available. So here photorespiration is absent. Thus, the basic pathway that results in the formation of the sugar, the Calvin pathway, is common to C3 and C4 plant. Did you note that the Calvin pathway occurs in all the mesophyll cells of the C3 plant? Yes, right? In the C4 plant, it does not take place in the mesophyll cell. Yes, it does not take place in the mesophyll cell because in mesophyll cell, rubisco is absent, right? But does so only in bundle seed cell? Yes, it does in bundle seed cell. Why? Because rubisco is present in bundle seed cell, right? That is here. Now this figure, first we understand this figure. What is given here? From atmosphere, look, from atmosphere CO2 comes. CO2 bind with H, look here, HCO3. It is converted into HCO3. How it converted? This CO2, it binds with H2O and forms H2CO3, right? And this H2CO3 is further broken down into H plus and HCO3 minus. This HCO3O minus, HCO3 minus is here, and it is binds with phosphoenol pyruvate PEP, right? PEP. And now what forms? This carbon fixation after what happens? C4 acid, and this C4 acid is oxaloacetic acid, right? Now this oxaloacetic acid, it is converted into which form? Malic acid or aspartic acid, and this malic or aspartic acid is transported to C4, that is C4 acid, malic or aspartic acid, right? When it comes into through plasmodesmata, look here, these are the tube-like structures, these are plasmodesmata. Now through decarboxylation, here decarboxylation occurs, means CO2 is released. When CO2 fixed, it is called carboxylation. When CO2 is released, it is called decarboxylation. What happens? One molecule of carbon dioxide is released, and this molecule of carbon dioxide, it go into the Calvin cycle, means C3 cycle. And this C3, and what happens here? Pyruvic acid is formed, and this pyruvic acid is transported back to the mesophyll cell. This one is our mesophyll cell, and mesophyll, and this one is our bundle sheath cell, right? Sheath cell. And this C3 cycle means what? This C3 acid means what? It is pyruvic acid, right? It is converted into phosphoenol pyruvate, that is regeneration. Here it used 2 ATP, right? That's why it is. How many ATPs are used? 3. In this 3 ATP in Calvin cycle, and this 2 means 5 ATP are used here. That means in one cycle, how many ATP is used? In one C4 cycle, how many ATP is used in? It is 5 ATP. And for the formation of one glucose molecule, it used how many? 30 ATPs. And in C3 cycle, it is only 18 ATPs, right? I think you understand this one. If any doubt is there, you can ask, right? Now this is the last. CAM pathway is absent here. CAM pathway is not described here, right? Okay, I tell you first CAM pathway. Then okay, first we will read this one, photorespiration. Then last very shortly I can describe you. Let us try to understand of one more process that creates an important difference between C3 and C4 plant. Important difference, which difference? Because it is present, this fast photorespiration, it is present in C3 cycle, but it is absent. C3 cycle it is present, but it is absent in C4 cycle, it is absent. And we know very well, because mesophyll cell, they lack Rubisco enzymes in case of C4 cycle. So this photorespiration is absent in C4 cycle, but it is present in which one? Mesophyll cell of that is C3 cycles, C3 plants. That's why it is present in C3 plants. To understand the photorespiration, we have to know a little bit more about the first step of Calvin's pathway. First step of Calvin pathway, that is the first CO2 fixation step. This is the reaction where RUBP combines with CO2 to form two molecules of 3-PGA is formed, because when RUBP, which is 5 carbon molecule, it bind with 1 carbon dioxide, it becomes 6 carbon molecule. And because intermediate process, unstable structure is formed with 6 carbon molecule, and when it is split, it forms 2-PGA, right? 2 molecules of 3-PGA. This is catalyzed by Rubisco. RUBP plus CO2, it forms 2 molecules of 3-PGA. But in case of happen, Rubisco, that is the most evident enzyme in the world. Do you know? Right? Wonder why? Because this is present in all the plants. And we know plants are that's why it is most evident enzyme. Cellulose is the most evident homopolysaccharide in the world. Why? Because all the plant cells, they are made up of cellulose. That's why here. It's characterized by the fact that its active site can bind to both CO2 and O2. Hence, the name, it is given carboxylase and oxygenase. Can you think how this could be possible? It could be possible because in Rubisco, what happens? This active site, it is bind with both. So if concentration is more of, if CO2 concentration is more, means more carbon dioxide is bind with Rubisco. If oxygen concentration is more, CO2 concentration is low, then what happens? Oxygen can bind with empty active site of Rubisco. Another thing, Rubisco has much greater affinity for CO2. When CO2 and the ratio of CO2 and O2 is nearly equal, when it is nearly equal, more carbon dioxide will bind with active site because CO2 affinity is more, right? But imagine what would happen if this were not so, means it is not equal. So this binding is competitive. It is a competitive binding. Who, right? It is the relatively concentration of oxygen and carbon dioxide that determines which of the two will bind with this enzyme. If concentration of oxygen is more oxygen will bind. If concentration of carbon dioxide is more carbon dioxide bind. If concentration is equal, then also more carbon dioxide will bind, because affinity of carbon dioxide is more, right? In C3 plant, some O2 does bind with Rubisco. Every time some O2 they bind with Rubisco. That why all the Rubisco enzymes, they are not involved for carbon fixation, or we can say for the formation of sugar. That's why here production is less than C4 plants, right? And hence CO2 fixation is decreased, because some Rubisco they binds with oxygen. Here the RUBP, instead of being converted to 2 molecules of PGA, when it binds with oxygen, it forms 1 molecule of phosphoglycerate, 1 molecule of PHOS, phosphoglycerate, right? That is PGA, right? It is 3 carbon molecule and 1 molecule of phosphoglycolate, glycolate, which is 2 carbon molecule, 2 carbon and 3 carbon molecule, right? 1 molecule of phosphoglycerate and 1 molecule of phosphoglycolate, which is 2 carbon molecule. This phosphate in the pathway called photorespiration, in the photorespiratory pathway, there is neither synthesis of sugar, no sugar is formed, no NADP, ATP is formed, But here what happens? Once CO2 is released, that means once CO2, we have loss of one CO2, right? There is neither synthesis of sugar and also not sugars nor of the ATP. Rather, it is results in the release of CO2. CO2 is also released here, loss of one CO2 with the utilization of ATP. In the photorespiratory pathway, there is no synthesis of ATP, no NADPH is formed, no sugar is formed. That's why the biological function of photorespiration is not known yet. What is the importance of this photorespiration for plants? It is not known yet, right? Because we know it is a waste process in plant. In C4 plant, photorespiration does not occur because rubisco is absent in mesophyll cells. This is because they have a mechanism that increase the concentration of CO2 at the enzyme site. Enzyme site means in bundle seed cell. In bundle seed cell, only carbon dioxide is released by malic acid or aspartic acid. But carbon dioxide is not reach up, oxygen is not reach up to, no gas, that is no free gas reach up to the bundle seed cell because they are thick layer and they are also totally impervious to gaseous exchange, right? Only carbon dioxide is go there. This carbon dioxide, it that means reach up to bundle seed cell with C4 molecule, that is with malic acid in the form of malic acid or aspartic acid, right? This takes place when the C4 acid from the mesophyll cell is broken down in the bundle seed cell to release CO2. This results in increasing the intracellular concentration of CO2. Continuously only CO2 is released. So what happens? Concentration of CO2 is released, oxygen is not, right? In turn, this ensures that the rubisco functions as the carboxylase, minimize the oxygenase activity. No oxygen means no oxygen, rubisco enzyme is not bind with oxygen. Maximum enzymes they bind with oxygen only. Now that you know that the C4 plant lack photophosphorylation. C4 plant lack photorespiration, right? Due to the absence of oxygen in bundle seed cells. And in mesophyll cell, due to the absence of rubisco enzyme, no photorespiration occurs. You probably can understand why productivity and yields are better in these plants. We understand because in addition, these plants show tolerance of high temperatures because we know here low temperature sensitive enzyme is present, which is called PPDK. It is called pyruvate phosphate dikinase enzyme. It is sensitive against low temperature. In low temperature, their productivity is less. High temperature, their productivity is high. Based on the above discussion, can you compare plants showing C3 and C4 pathway? We will compare this one. Use the table format given the table and fill the informations. We will fill this one, right? Now come to the table, this table. In this table, fill in the columns second and third in this table to highlight the differences between C3 and C4 plant. What are the difference between the C3 and C4 plant? Here we can understand this one, right? Through this table. What is given here? Characteristics and C3 plant, and choose from this one. Cell type in which the Calvin cycle takes place. We know in C3 plant, which one? Mesophyll. It is mesophyll. Right? And in C4 plant, what? Bundle seed. In C3 plant, cell type in which the Calvin cycle takes place. This Calvin cycle takes place in mesophyll, right? MESO. And in case of C4 plant, it is Calvin cycle takes place in bundle seed cells. Correct? Cell type in which the initial carboxylation reaction occurs, it is in mesophyll cells, and here initial carboxylation reaction occurs where? It is in bundle seeds. Carboxylation means what? Carbon fixation. How many cell types does leaf have then fix CO2? Here only one, that is mesophyll. Only one. Here two, it is bundle seed and mesophyll. Right? That is two here and one here. Now, which is the primary CO2 acceptor in case of C3 plant? It is our RUBP. CO2 acceptor, RUBP accept the CO2 and forms 2 PGA. And in case of C4 plant, which one is? Phosphoenol pyruvate, right? Number of carbons in the primary CO2 acceptor. How many numbers are for carbon? RUPJ 5 carbon atom and PPJ 3 carbon atom, right? Another is given, which is the primary CO2 fixation product. Product is our this PGA in C3 plant and oxaloacetic acid. It is in C4 plants. Number of carbon in the primary CO2 fixation product. PGM is 3 carbon and OAM is it is 4 carbon. That's why it is called C4 cycle and C3 cycle, right? Then after, does the plant have Rubisco? Yes. It is always yes, because Rubisco is present in both plants. In case of C3 plant, Rubisco is present in which? It is present in mesophyll cells and Rubisco is present in bundle sheath cell, right? Does the plant have PEP case? No. And it is yes, right? Which cell in the plant have Rubisco? It is our mesophyll cell and here it is bundle sheath cell, right? CO2 fixation rate under high light conditions, CO2 fixation rate under high light condition, right? Low, high or medium, it is given. It is high light condition means what? Low, and here it is high, right? Because in high light condition what happens? Here photorespiration occurs, right? Now whether photorespiration is present at low light intensity, negligible. Low light intensity means no. Here also it is absent. Photorespiration is totally absent. Whether photorespiration is present in high light intensity, yes, high, right? But in case of C4 plant, because photorespiration is absent, negligible. Whether photorespiration would be present at low CO2 concentration, if concentration is CO2 is low, yes, it is high, right? But in case of C4 plant, negligible because photorespiration is absent. Or we can say which one? Rubisco enzyme is absent in mesophyll cells. Whether photorespiration would be present at high CO2 concentration, if CO2 high concentration, this CO2 concentration is high, then photorespiration would be high? No, negligible. And here also it is negligible, right? Because high concentration means this Rubisco binds with only which one? Carbon dioxide. Temperature optimum, it is low temperature means 20 to 25 degrees centigrade. It is high temperature means 30 to 40 degrees centigrade, right? Examples means like we have wheat, rice, tomatoes, other things are also there. And in case of C4, here means sugarcane, sorghum, amaranthus. These are C4 plants, right? So through this table, we can understand what is the difference between C3 and C4 plant, right? Now another point here we have to describe, that is factor affecting photosynthesis. What factors which are responsible for the affecting of photosynthesis, right? Now two types of factors are there. One is called internal, which is present inside the cell, inside the plant, and another one is called external factors. External means we have 4 factors, that is light, second one is our carbon dioxide, third is temperature and water. Okay, look here what is given. Understanding of the factor that affect photosynthesis is also necessary. The rate of photosynthesis is very important in determining the yield of plant, including crop plants. Photosynthesis under the influence of several factors. What several factors is given here? First given here, look, internal as well as external. The plant factors include the number, size, number of leaves, number of size, size of leaf, age of plant, orientation of leaf is phyllotaxy of leaves, mesophyll cells, right? And chloroplast, number of chloroplast, they are granular or granular, internal CO2 concentration and the amount of chlorophyll. These are the internal factors which are totally genetic predisposition, genetic, and also they are adaptation according to the plant. We can say growth of the plant, because we know if it is a dehydrated plant like acacia, leaves are very small, right? Very small leaves means what? Photosynthesis, this is size. Photosynthesis rate is also decreased, right? Now external, these are internal. Now external factors would include the availability of sunlight. First sunlight, second one is our temperature, third one is our CO2 concentration, and fourth one is our water. 4 things are given: sunlight, temperature, carbon dioxide and water, right? Concentration of carbon dioxide. As a plant photosynthesizes, all these factors, whether it is external or internal, simultaneously affect its rate. Hence, those several factors interact and simultaneously affect the photosynthesis or CO2 fixation. Usually, one factor is the major cause and is the one that limits the rate, and this is the factor which is near to the limit. It's a minimal range, right? That I explained you like this one. Suppose that we have that means a sufficient amount of water, chlorophyll is also there, sunlight is also present, but in the absence of carbon dioxide, means what happens? When carbon dioxide finishes, means what happens? Formation of glucose is stopped. When we provide this glucose, then this reaction starts. So which one is limited? Which one becomes the limiting factor? It becomes carbon dioxide the limiting factor, because in the absence of carbon dioxide, this cycle is stopped, right? So who is limiting? Which one in its minimum range, right? That is given here also. Hence, at any point, the rate will be determined by the factor available at suboptimum level, right? And who gives this limiting factor law? Law of limiting factor is Blackman's, who gives in 1905 the law of limiting factor. According to this one, what is this factor? What is this limiting law of limiting factor? It is that this states the following: if a chemical process is affected by more than one factor, then its rate will be determined by the factor which is nearest to its minimal value, which is nearest to is the minimal value. That I explained you. If CO2 absent, CO2 means it is the minimal range. CO2 absent means stop. When we provide CO2, it starts, right? It is the factor which directly affects the process if its quantity is changed, right? So minimum substance becomes limiting factor. For example, despite the presence of green leaf, optimal light and CO2 condition, the plant may not photosynthesize if the temperature, here example is given temperature is very low, because we know C3 plant 20 to 25 degrees centigrade and C4 plant means it is that is 30 to 40 degrees centigrade. If temperature is low, means it is below than 0 degrees centigrade. What happens? No, enzymatic reaction stops. What happens? No photosynthesis here. That means what? At that time, temperature will be limiting factor, right? This leaf, if given the optimal temperature, it will start photosynthesis. That what happens? This temperature becomes limiting factor. Okay. Now here these four factors. First is light. We need to distinguish between the light quality, light intensity, and duration of exposure of light, right? While discussing the light as a factor that affects the photosynthesis, it is a very rare. Light as a limiting factor is very rare. It is present only shady places or dense forest, right? There is a linear relationship between incident light and CO2 fixation rates at low light intensity, right? What is given here? Linear relationship. At higher light intensities, gradually the rate does not increase, right? The rate does not further, as you say, you can say in this that is a graph. After some time, what happens? Efficiency is stopped. Now it becomes constant, right? Light intensity is increased. First, it is increased. The rate of photosynthesis is also increased, but after some time, what happens? It becomes constant, right? At higher light intensities, gradually the rate does not show further increase as other factors become limiting. At that time, what happens here? What is interesting to note that the light saturation occur at 10% of the full sunlight, only 10% of the full sunlight. Hence, except the plant which grow in shade or which grow in dense forest, right? Light is rarely limiting factor in nature. Increase in incident light, or one thing also, when we increase the incident light or intensity of light, what happens? It also destroys the chlorophyll. That is called photolysis of chlorophyll. Means it is responsible for, at that time it is responsible for photolysis of chlorophyll, and photolysis of chlorophyll means breakdown of chlorophyll. That means what happens when chlorophyll damage, no photosynthesis occurs. That means it decreases the photosynthesis, right? This one is called PHOTO, photolysis, photolysis of chlorophyll, right? So photolysis also, that is harmful for chlorophyll. When incident light beyond a point, that is okay. Now second is carbon dioxide concentration. This is the major factor, right? Carbon dioxide is the major limiting factor for photosynthesis. The concentration of CO2 is very low in the atmosphere. It is between 0.03 and 0.04%, right? When we increase the concentration up to 0.05, that the carbon fixation is also increased. At that time, it causes the increase of CO2 fixation, right, rate. But beyond this, the level can become damaging over a long period. But when we increase the concentration beyond the 0.05 for a long time, what happens? It damages the plant, right? The C3 and C4 plant respond differently to CO2 concentration, right? At low light condition, neither group respond to high CO2 conditions. At high light intensities, both C3 and C4 plant shows increase in the rate of photosynthesis. They also increase the rate of photosynthesis. What is important to note that the C4 plant shows saturation at the level of what? 360 micro per liter, right? While C3 respond to increase the CO2 concentration and saturation is seen only beyond of 450 micro per liter. Look here, what is thing? Sometimes you get confused, but you can understand. C4 here it is given 3, so C4 360. C3 here is given 4, just opposite 4, so 3, it is C3, right? 450 micro. Thus, current availability of CO2 level is limiting to the C3 plant, right? The fact that C3 plant respond to higher CO2 concentration by showing increased rate of photosynthesis leading to higher productivity also has been used for some greenhouse plant in polar region. What happens? They form say glass house, right? That is called greenhouse crops such as tomato and bell pepper. They are allowed to grow in carbon dioxide enriched atmosphere that leads higher yielding. So C3 plants means they show high productivity in high CO2 concentration. Why the high concentration? Because when high concentration is there, then RUBP binds with CO2 mostly, right? And more productivity is there. But in case of C4, what happens? C4 means CO2 concentration is already they bind with only CO2, not with not with oxygen, right? Temperature, the dark reaction being enzymatic, we know enzymatic, and enzyme is it has an optimum temperature. Are temperature controlled? Though the light reactions are also temperature sensitive, they are affected to much lesser extent, but they affect, right? The C4 plant respond to high temperature and show higher rate of photosynthesis, while C3 plant have a much lower temperature optimum, right? C4 means higher rate of photosynthesis, while C3 plant have much lower temperature optimum, right? 20 to 25 degrees centigrade, and C4 means 30 to 40 degrees centigrade. The temperature optimum for photosynthesis of different plants also depend on the habitat they are adapted to, right? Means it is polar or it is temperate. Here given tropical plants have a higher temperature optimum than the plant adapted to temperate climates. You understand this one? Water, even though water is one of the reactant in the light reaction, the effect, because in light reaction what happens? It is due to photolysis. What happens? Water is split into H plus and OH minus, means oxygen is evolved from water. First thing is the electron is also evolved from water. Another H plus, that means H plus is also evolved from water. That is very important in light reaction in case of non-cyclic photosynthesis, right? The effect of water as a factor is more through its effect on the plant rather than directly on photosynthesis. Water stress causes, it is not right directly, indirectly it is. Water stress causes the stomata to close, hence reducing the CO2 availability. Besides, water stress also makes leaf wilt, thus reducing the surface area of the leaves and their metabolic activities as well, right? When stomata close, means what happens? What happens? Exchange of gases not occurs, right? So these are the some factors which is given in our NCERT. Now this chapter is completed, but something I want to tell you right before close this one. Look here, this is our C4 cycle and this side it is CAM part. What happens here? In C4 cycle, I already tell you because we have a less time. CO2 comes from our atmosphere and it binds with CO2 plus H2, it forms H2CO3. H2CO3, you already know this is the carbonic enzyme, right? Carbonic acid. Now H2CO3 is broken down H plus and HCO3 minus. This HCO3 minus, it binds with phosphoenol pyruvate and forms oxaloacetic acid, which is a 4-carbon molecule. 4-carbon molecule. PP is a 3-carbon molecule, right? Now this oxaloacetic acid, by using a NADPH2, it converts into aspartic acid or malic acid. It is also a 4-carbon molecule. When it is converted, so look, aspartic acid, malic acid, it is converted inside the mesophyll cell, not in bundle cell. These reactions occurs inside the mesophyll cell. Now through plasmodesmata, because it is totally airtight chamber, it is thick wall and it is impermeable to gases also, right? Bundle C cells are. Now this aspartic or malic acid, it is transported to the bundle C cell through plasmodesmata. When it comes into the bundle C cell, what happens? Through decarboxylation. Decarboxylation means what happens? CO2 is released. They release one CO2 and this CO2, it comes into the C3 cycle, Kelvin cycle. It becomes a part of Kelvin cycle. And because they release one carbon dioxide, now they becomes once more 3-carbon molecule and that is our pyruvic acid. When they convert into pyruvic acid, this pyruvic acid, it is transported or returned back to the mesophyll cells. In mesophyll cell, this pyruvic acid is converted into phosphoenol pyruvate by using of 2-ATP, right? That means what? Here in mesophyll cell, here 5-ATP is used. 3 in Kelvin cycle and 2 in mesophyll cells, right? Now in second, look here, this is the CAM pathway which is not in our NCERT, but some question will come here. These both are C4 cycle. It also shows C4 cycle, but it is called as CAM pathway. What is the difference between these two? Here 2 cells are involved, mesophyll cell and that is bundle cell cells, right? Here Rubisco enzyme is present. And this side, PEPcase, PEPCASE, phosphoenol pyruvate carboxylase enzyme is present. That is right. But here in a single cell, in CAM pathway, single cell, this CAM pathway it is present in. It is also called as crassulaceous acid metabolism. It is present only in which plants? Succulents, xerophytic plants. Right. Here a specific type of stomata is present, which is open only and only in daytime. Night time, but absent in daytime, right? Okay, what I tell you here, a specific type of stomata is present because they are xerophytic plants. So they only open in night, right? Stomata open in night but close in daytime to minimize the water loss only, right? They also store foods. They have succulent. They are called succulent plants like aponcia, aloe vera, right? Bryophyllum, these are. And pineapple also, welwitschia also, right? What happens here? In night time, CO2 come from this stomata. This type of stomata is called spot active, SCOTACTIVE, spot active stomata. Specific stomata which close in daytime and open in night time to minimize the water loss, right? What happens? CO2 comes from atmosphere in the night, and by using of this PEPcase enzyme, that is present also in C4 cycle and forms oxaloacetic acid, which is 4-carbon molecule. This oxaloacetic acid is converted into malic acid, which is also 4-carbon molecule. Now this malic acid, it is stored in vacuole in the night because no photosynthesis occurs. In night, malic acid is formed and this malic acid is stored in vacuoles. In daytime, what happens? This malic acid is released from vacuoles and this malic acid through decarboxylation, here also decarboxylation occurs, it releases CO2, and this CO2 it becomes a part of C3 cycle. Means it is used in C3 cycle in Calvin cycle. Here also Rubisco enzyme is present. Rubisco. Both enzymes are present in the same cell, in mesophil cell, right? Pepcase and Rubisco both are present in same cell, right? Then what happens? This, after releasing the one carbon dioxide, this malic acid converted into pyruvic acid, which is 3 carbon molecule, and now this pyruvic acid is converted into PEP case by using of ATP to ATP, like to in C4 cycle. So this cycle it occurs in a single cell in case of CAM pathway, right? So CAM pathway, here it is given. CAM pathway is called crassulation acid metabolism because in night time acid is formed and stored in vapor. In daytime this acid releases CO2, which involves in photosynthesis, right? Okay, now next, photorespiration. So photorespiration, some information is given in our NCERT, but a cycle is not given. In photorespiration, what occurs? It is also called a C2 cycle. Here, 3 types of cell organelles are involved. It is occurs in mesophil. Mesophil means in which mesophil? C3 plants. In C3 plants. C4 plant, this photorespiration is absent. What cell organelles? Chloroplast, peroxisome, and mitochondria. Look here. This is chloroplast. This is peroxisomes and mitochondria. What happens here? Oxygen, because here carbon dioxide concentration is low, or we can say it is high in high temperature or high light intensity is there. Then what happens? Oxygen binds with RUBP, right? This RUBP and forms 2 molecules. One 3-PGA, which is 3 carbon molecule, and one phosphoglycolate, which is 2 carbon molecule. Now this phosphoglycolate it forms a ATP and convert into glycolate. This glycolate, when it is converted into glycolate, what happens? This glycolate is transferred into the peroxisomes, like here. In peroxisomes, because they have oxygen molecules, so this oxygen binds with glycolate and forms glyoxylate. Glyoxylate, by using a amino group, it converts into glycine. Glycine, which is a amino acid. You know very well, it is the simplest glycine, right? And it is 2 carbon molecule. This glycine is transported into the mitochondria. 2 molecules, or we can say 2 moles of glycine, right? What happens? They bind with each other, and what happens here? Through decarboxylation, one carbon dioxide is released, and this carbon dioxide is not used in photosynthesis, but it is released into the atmosphere. That is loss of one CO2, right? One NH2 is also released, and now a 1 molecule of serine is formed. Serine means it is 3 carbon molecule, right? Because here 2 glycine means 2 molecule of glycine means it becomes 4 carbon molecule. 1 carbon is released in the form of carbon dioxide. Now remaining 3 carbon, they form serine, right? Serine. Now this serine is transported into once more to peroxisomes. What happens in peroxisomes? They also release NH2. The second amino group is also released. Now it is converted into hydroxypyruvate, right? Now hydroxypyruvate, it is by the form of NADP, converted to NADPH, right? Now OH is released. Now it becomes glycerate. It is converted into glycerate. Hydroxypyruvate is converted into glycerate, right? Where in peroxisome. Now this glycerate means what? Glyceric acid. It is transported into chloroplast. And in chloroplast, by using ATP, because here 1 ATP is formed, but this side what happens? 1 ATP used means net gain means 0, right? So this glycerate, it is converted into phosphoglycerate, or we can say phosphoglyceric acid, which is a 3 carbon molecule, right? And now this phosphoglyceric acid is converted to phosphoglyceraldehyde, and through regeneration, we can say here through regeneration, it is converted into RUBP, phosphoglyceric acid. Then after it becomes phosphoglyceraldehyde, and then it becomes RUBP. So this is a cycle in which 3 cell organelles are involved. This is called which is C2 cycle or photorespiration, which is a useless process in plants. Okay. Now I think everything we discuss here about the photosynthesis, right? Nothing will remain here. If you find out any doubt, you can ask. Okay. Thanks.